Hi, my name is Jack. I am life sciences, and this is my BlueSky proposal. So I was focusing my project on osteoporosis because osteoporosis is a bone metabolic disorder that affects millions of people in the United States and around the world. So the United States population is becoming older, and as generations pass, this disease is going to become more prevalent. It doesn't really have a cure right now. It only has like treatment options, and what we want to do is actually find the genes that affect osteoporosis, so that way we can cure the disease. Osteoporosis is a bone metabolic disorder that is becoming more prevalent in the United States, and right now there is no specific cure for it, only treatment options, and what we want is to have a cure for it so that way it costs the United States less money and we can help millions of people who are suffering from this disease. I proposed a two-step approach. The first step was identifying the genes that affect osteoporosis and the ones that most greatly contribute to the disease, and the second step was seeing how those genes work together and interact to cause the disease. For the first step, I basically proposed to do a microarray study, which would actually so microarray is basically a molecular technique that allows you to determine which genes are upregulated and downregulated between two different types of cells. So the two different types of cells I would be studying would be osteoporotic cells and regular bone cells. So that would tell you which genes are more upregulated or downregulated in osteoporosis tissue compared to regular tissue. And then after we would find those genes, because microarray usually generates a lot of data, you would want to sort through that data and quantify the genes that you're studying, like the gene expression. So to do that, you would have to use qPCR, which is a more accurate PCR technique, and that will let you quantify the genes that are most upregulated or downregulated, and then that would allow you to pick the most important genes out of that pool of genes that you would have. The second step of the approach is actually finding the relationships between the genes, and by what I proposed was doing something called gene set enrichment analysis, basically a computer software that allows you to compare gene expressions among a group of genes that you're studying and try to figure out where exactly in the cell those genes are being expressed and how they relate to each other. And then the final step of the second half of the approach, the second part of the approach, would be to actually do in vitro studies on, like, let's say like mice or any like mammal. So I try to include things that we learned in life sciences, molecular techniques, or closely related molecular techniques such as PCR. We learned PCR in Life Sciences 1A, and I used qPCR, which is called quantitative polymerase chain reaction. So it's very similar to the polymerase chain reaction we learned in class.